Hello and welcome to my next live stream. This is on F211 Unit 1 AS Biology. So as usual, are the audio levels and quality fine? Is the camera fine? Everyone happy for the next hour and a half? And then there'll be the Unit 2 F212 live stream starting at 8 o'clock till 9:30. Right, well, does anyone have any questions to start with? If not, I've got some preset topics to do for the AS. Well, that's convenient, Jessica, because someone's already asked me to do that. So yes, I will definitely go through that. Then I think that's a good place to start. Right. So carrying of hemoglobin, how it transports oxygen and carbon dioxide. Well, hemoglobin is a globular protein which contains heme groups. Now, these heme groups contain iron, and the iron can bond with oxygen, which means hemoglobin has an affinity for oxygen. And the ability, that's what affinity means, the ability of hemoglobin to take up and release oxygen. And it depends on the surroundings. So if there is a high partial pressure of, well, we look at the pressure, partial pressure of oxygen. And what does it will take up, when it's got no oxygen attached to it, it will quite easily take up oxygen. And when it's got full oxygen, it obviously will not take up any because it can't. But equally, at low partial pressure of oxygen, there's not much oxygen. It won't readily take up oxygen. Now, this is because if we look at, I'm going to just draw it with one heme group. If this is the kind of globular protein of hemoglobin, the heme group will be in the center. Now that means it is quite hard for oxygen to become attracted to. So it needs a high partial pressure, which will then cause one oxygen to join, which will then change the shape. So I'm going to draw it. Heme's there. Now this will change the shape of oxygen of hemoglobin, to make it easier for oxygen to get in. In this case, I've just let it get in there. Now, if it can do, that means it can attract the next two molecules quite easily. But after the third molecule, the shape changes again, so it becomes a lot more closed, making oxygen harder. So it's hardest for the first oxygen to join and the fourth oxygen to join. But after the first oxygen joins, it changes shape, so the second and third can join quite easily, but it changes shape again, so the fourth oxygen molecule struggles to join because it can only attract four. And that's why you get partial pressure of O2 and percentage saturation. That's why you get this kind of S-shaped curve. So that means the partial pressure needs to increase a lot to attract the first oxygen Then only a small amount increase will allow it to attract the next two, and then a much bigger increase to attract the fourth. So that's how it picks up oxygen. Does that bit make sense so far? There's the picture. Jessica, does that make sense? Okay, good. Now, carbon dioxide. Now, there are three ways carbon dioxide can be transported. 5% is dissolved directly in blood plasma. Blood plasma. Sorry. 10% is combined with hemoglobin to form something called carbaminohemoglobin. So you can have hemoglobin combined with oxygen to form oxyhemoglobin, but you can also have carbon dioxide form with hemoglobin to form carbaminohemoglobin. But 85% of transported is hydrogen carbonate ions. So we're going to look at how that happens. You get carbon dioxide plus water. And this happens in a red blood cell, which I'm just going to draw like this. Red blood cell. So we have carbon dioxide diffuses into the red blood cell, and it combines with water in the red blood cell to form where is it? Yes, good. It forms a carbonic acid, which is H2CO3. So you can see the oxygen, CO3, H2, and a carbon. And this is using the enzyme carbonic anhydrase. So we then have carbonic acid. Now, acids very often do something called dissociate. So that means you kind of lose an H plus or a proton. So this dissociates to form H plus HCO3 minus. And that is a hydrogen carbonate ion, HCO3 minus. And then what happens is you then have a increase in H plus, and that will join with hemoglobin, which is Hb, to form hemoglobinic acid. Now, the hydrogen carbon ions, diffuse out. And what this happens, it creates a increase of negative charges outside the cell, and then there is a decrease negative charges inside the cell. Now, so to make it balance out, chloride ions will then diffuse in. It basically means that you don't have an increase of negative ions inside and outside the cell. It's all remains, let's say you have 10 minus ions here and 10 minus ions out here. When these ones move out, you get the same amount moving in, so it's you don't lose charge. So just to go over that, 5% is dissolved directly in plasma, 10% is combined with hemoglobin to form carbaminohemoglobin, just like oxygen combines with hemoglobin. And then the rest, the other next 85%, combines with water in the red blood cell to form carbonic acid, which will then dissociate to form a proton and a hydrogen carbonate ion. The proton can form with hemoglobin to form hemoglobic acid. The hydrogen carbonate moves out and chloride moves in to make sure the charge balance doesn't change. And then this carbon dioxide will cause oxygen to be released because hemoglobin's got a higher affinity for carbon dioxide than oxygen. So you have lots of carbon dioxide, then oxygen is released because hemoglobin with 8 oxygens, so oxyhemoglobin, will become and then this hemoglobin can then combine to form carbaminohemoglobin. So does that make sense? Right, excellent. Does that cover everything that you wanted then? And if so, what's the next question people have got? I was explaining about car transport of carbon dioxide. Oh, got a few questions. Right, give me 1 second. Right. Um. Oh dear. Right. Can you go mind? Um. Call me Jay. I went over translocation in the previous live stream. I don't know when during the previous live stream, but during the previous AS live stream, I did go over that. So if you want to have a look on that, the answer should be there. Or if you didn't like the way I explained it, I can go over it again. But if you can have a look for it there. May 2010 question. Um, Transport carbon dioxide unit one. That's why this is the unit one live stream. Um, I, I I asked for people to post before the live stream what questions, exam questions they wanted doing. Um, oh dear. Right, I'll have to load up the website then. All right. Please, for the unit two one I'm doing later, please can you post the questions from actual exam papers before the stream? It makes it a lot easier than me having to search them up during the live stream. Call me Jay. I will go over it if you do want me to, but can you check the previous live stream first? Just I don't want to repeat myself. Um, Ezra, I personally think it's going to be blood vessels because the heart came up in January last year, but we'll see. Right, May 2010, question 4B. Uh, 10, question 4B. What's this on? Oh, meiosis. Right. Right. Identify one pair of homologous chromosomes. Right. Yes, I did see your question about receptors. Don't worry. I'm just answering them in order. When you're doing chemistry, the simple answer is I'm not, because people wanted to do these in units, and I simply cannot do one on chemistry as well. What I suggest is if you ask your chemistry, if you have just one chemistry question, ask me it now, and I will see if I can fit it in during the live stream. Right. Where am I? May 4B. Right, I'm a bit confused on this question. Right. Identify one pair of homologous chromosomes. Homologous chromosomes is literally a pair which are the same. So there are 6 chromosomes. There is 2 of them that are short ones, 2 which are long ones, and 2 which have half of them very long, half of them very short. Those each of the 2 that look very similar. That's a homologous pair. So they want you to circle one of them. So you circle the 2 short ones, that's a pair. You circle the 2 long ones, that's a pair. Or you circle the 2 half short, half long ones. Right. And I. Alright. The nucleus of sperm is produced in meiosis. Draw a diagram supposed to represent the chromosomes that are present in the nucleus of a sperm from the same animal. If I'm not very much mistaken, it must be half the number of chromosomes. June 2010. Right, yeah. So what you do is meiosis will produce half the number of chromosomes, and what you do is so only do one from each pair, so one short one long one, and one half long, half short. But also you have to cut the chromosome in half. So if you have chromosome looks like this, it will cut in half. So you have to draw like that, because during meiosis it will you get the pair splitting in half, and then the chromosome splits in half as well. I hope that answers your question. Um, right. I'm doing them in the order of which I'm asked. Right. So the role of membrane-bound receptors. In the book, it's stuff like membrane-bound receptors. Right. Well, receptors are molecules which have a complementary shape to a specific molecule and combine to it, usually a hormone or another chemical. Now, these receptors are generally protein molecules, and I'm going to look at. Oh yeah, I can see what you mean. Yeah, insulin receptors, medicinal drugs, and hijacking receptors. Um, I'm telling you that I've never really seen much asked on this. They will generally ask the role of receptors, but receptors will be something like glycoproteins or glycolipids. They can act as receptors, or you can have different proteins for receptors themselves. Yeah, you just need to. Basically, I don't think these are just three examples they've given here. So what you could have is right. You have a cell. You have a receptor. This is a very big receptor. You then have, let's say, a hormone molecule come along and combine perfectly because it's a complementary fit. This will then cause, an internal change in the cell to occur. So perhaps another molecule, because this molecule cannot enter the cell, so this will cause another molecule to be made. Let's draw it here. It will then go to the nucleus and cause it to have effect. So it might activate a protein or produce something to be made. Medicinal ones are just ones which are, you create a drug which you know will be complementary to the cytoreceptor and will cause an effect. And hijacking receptors are viruses that can enter cells. They can bind with the receptors on the cell plasma membrane and will then kind of enter the cell that way and can take over the cell. Those are three examples. But basically, you just need to know you have a molecule complementary to the receptor and it causes an effect to happen, so something to change inside the cell. That's what you need to know. And you need to know that it's a protein molecule like a glycoprotein or a glycolipid. That's not a protein, a glycolipid, but yes. I hope that answers your question. Question 3BI on Jan 2012. Where's it? 3B. So I'm just trying to get the question. Which one was it? Was it 3BII? Describe and explain how the wall of an artery is adapted to both withstand and maintain high hydrostatic pressure. To withstand pressure and to maintain pressure. Right, to withstand pressure. So the obvious one is the wall is thick. We're always talking about thickness of the walls. And you've got the layers of collagen. And collagen, as a structural protein, always provides strength. So those are three I would say. It's a thick wall with collagen in it, which can provide strength and support to the artery. So that's what helps it withstand pressure. Now, what helps maintain pressure has got elastic tissue, which can stretch and then cause recoil. So it stretches. One sec. You'll stretch, go out, and suddenly recoil, which decreases the volume, so it means that the pressure will be suddenly increased and will give it a little boost. And also you have smooth muscle, which can constrict the lumen, yet again reducing the volume, so it increases the pressure. So I hope that answers your question. Where am I next? Can you go over how to answer biodiversity and evil? Aubrey, the best way I mean, how to answer those questions is exactly the same how to answer all other questions. If you can give me some examples, I will go over that. Also, that is Unit 2, which I will be doing at 8 o'clock. So if you can send me some examples now on my Unit 2 live stream, I will then answer them during it later between 8 and 9:30. How tissue fluid is formed and lymph. Brown like Brownlee, on one of the previous live streams, I have mentioned how tissue fluid is formed. I believe that was on the first live stream, so the general live stream, if you can have a look there. Lymph is just literally when the tissue fluid drains away into the lymphatic system. That's all it is. So just basically, blood is when it goes into the tissue fluid. That's when blood goes into the cells around, the liquid around cells. That's tissue fluid, and then when it drains down into the lymphatic system, it's called lymph. That's basically how they are formed, and I will quickly explain this, but do look on the previous, I think the first live stream. All right, the arterial end starts decreasing in volume to the capillaries in blood vessels. This increases the hydrostatic pressure, which causes blood to be pushed out, and that's tissue fluid. But then at the venual end, the pressure had decreased slightly, but you have a water potential gradient because there's a lot of proteins in the blood, but there's a lot of, just pure liquid water in the tissue fluid. Yeah, there's a lot of water in the tissue fluid, which causes the tissue fluid to return back into the blood. Some of it, though, is drained away into the lymphatic system. That is it very briefly. So that is basically it. So you can look it up again in the previous live stream. Can you cover these two things? Right. Describe the cardiac cycle reference to the action of valves in the heart. I'm not going to go through it incredibly slowly because I've got a lot to do, but you can pause this and re-watch bits or watch it later to make notes on it. And again, it is done in one of my videos if you need to, but I will go over it. Right. So we can start at, you have atrial systole, ventricular systole, and then diastole. So those are the three main stages. And the stages are atrial contraction. So the heartbeat starts when the atria contract. Both the right and the left atria contract together. And this causes blood to flow from the atria to the ventricles. I can't draw a very good heart. But the contraction of the atrium causes blood to be pushed from the atrium into the ventricles. And what happens is, because I'll draw this bit of the heart. Here's an atria, and here's a valve. Let's say this valve is closed. And that's the ventricle. As these contract, cause pressure to increase, causes blood to go through, push open. I'm going to draw the next one. That's the ventricle. And you have, so pressure increases, pushes on the valves, causing them to open, and blood flows through into the ventricles. That's atrial systole. Now, ventricular ventricular systole is very similar in the fact that it's very similar in the fact that the ventricles contract, which push blood through here and causes the vessels again to open. Now, those vessels are the semilunar valves, or the valves, sorry, not vessels, valves. And these are the atrioventricular valves. But also, the blood will push back on these valves, which cause them to close. Because it's kind of like that, so they can be pushed open. If blood pushes back on them, they close. So no blood can go back. So yeah, so pressure increases because these contract, push back on the atrioventricular valves, causing them to close so blood can't flow back, and pushes on the semilunar valves, causing them to open and flows into the appropriate vessel. Then, yeah, and then you have diastole, which is basically kind of like the rest period, and you just basically blood is going to push back on these valves, the semilunar valves, which will cause them to close, and you then get blood starting to flow into the atria again. And that's the cardiac cycle. Then you said you wanted SAN, wasn't it? Control of the cardiac cycle. Describe how heart action is coordinated. Right. Again, I'm going to draw a really bad diagram. Right. I say this is not meant to be a good diagram. That's the apex. Purkinje tissue. Right. Sorry. Right. SAN, AV. Okay, AVN should be a little bit more over there. I'll just get the idea. Right. So an excitation wave is formed from the sinoatrial node, which will spread through the tissue until it reaches the wall between the ventricle and the atria, or between the atria and the ventricle. It then goes into the atrioventricular node, where it is held, because these walls here are insulating. They cannot conduct electricity. So it's a disc of tissue that cannot conduct the wave of excitation, but it can go in through to the atrioventricular node, which holds it for about 0.1 second, I believe. And then after that, it releases it again, where it goes down the Purkinje tissue to the apex, and then the wave goes upwards, causing the ventricles to contract from the base down. So SEN releases wave of excitation, which goes along the walls of the atria. There's a non-conductive wall between the atria and the ventricle, so it can only go through the atrioventricular node, which holds it for a short period of time before sending it down the Purkinje tissue to the apex, which then causes it to move up the ventricles, pushing blood upwards and out of the heart. I hope that answers your question. Right. I'll answer the first question later because I did say I'll do it in the correct order. Can you go how water is transported by xylem? So that is, right, I saw something. Aubrey said, and the process of transpiration and translocation. I said earlier, I've done translocation in a recent live stream. I can't remember which one it was, but if you look either at the AS one or the overall one, I have done it there. So can you look at that so I don't have to explain it again just on this video for time reasons. But I will go over transpiration. I will do that. If you saw the last live stream and didn't understand translocation, I'm more than happy to go over it again. But I haven't done translocation before, so transpiration before, so right. I'm just going to ask now, for transpiration, do you understand do you want just a transpiration stream, or do you want how it all moves in the root and in the stem? So like adhesion and cohesion and tension in the stem and the apoplast, symplast and vacuolar pathway in the roots. Do you want that as well? Just answer that, then I'll go through it. In the meantime, though, while I wait for your answer on that, I will start with the I will do the structure of xylem and phloem. Right. Okay. I'm just going to explain actually. Right. Xylem. You have xylem vessels, which will carry water and minerals. These are long, thick. These cells have long, thick walls, which have been impregnated by lignin, which is a supporting feature. The lignin also waterproofs, provides strength and support. Yeah, things like that. Now, these cells are dead, so they're long, thin cells. I will draw it. Long, thin cells. They have no end walls. So there's lignin here. No end walls, so water can pass between cell. So cell. No end walls. And there's lignin in the wall. There is no contents. The cells. There's nothing in the cells. So you don't have a nucleus and all these different things. So xylem vessels are dead. Because lignin waterproofs the walls of the cells. As a result, the cells die and their end walls and contents decay. So you get no things in the cells. So it's a dead, just to allow water passage to easily go through it. In some places, lignin isn't complete, so it might leave a pore, so water can leave or water can enter the cell. So that's it. It's basically long cells, no end walls, dead, so no organelles in it. Lignin to strengthen support, prevent from collapsing, and waterproof the cell, and pits in the lignin to allow water in and out. Yeah, that's xylem. You then have phloem, which has two cells. You have a phloem sieve tube and a companion cell. Again, a phloem sieve tube has no organelles, so no nucleus, no mitochondria. The companion cell has all that, so nucleus, mitochondria, endoplasmic reticulum, like that. So the companion cell has all the contents that make it work. So many mitochondria produce energy, has a nucleus, so that's what keeps this cell alive. Each sieve tube has a companion cell to go with it. And they do contain cytoplasm, but not very much. They're lined end to end again, and they have the tube does contain cross walls in intervals, so like this. So it contains like holes they contain walls, but these are perforated, we call them, with many pores, so you can allow things to go through. So that's basically what it, and then the gaps here are called plasmodesmata that allow things in and out between the sieve tube and the companion cell. Thank you for the comments on the chemistry videos. Literally, I spent all day doing videos. Now I'm doing live streams. Wonderful. Yeah. Anyway, so these are plasmodesmata, long tubes, perforated ends to the walls to allow things to go through. Companion cells contain mitochondria, nucleus, all the organelles to keep the cell alive. This cell has very little cytoplasm, no organelles, and transport sugars. Right. I saw right, brown like brownie, active and passive immunity I will go through, but that's unit 2. So I'll be doing that in the unit 2 live stream, which is later today. It will be 8 till 9:30, I said. So I will answer that question then. Which book? The official textbook. It is the official textbook. Always refer to that one as your final one. Um, right. So, um, can you explain how it simulates loading the flow? I've done that on a previous video. Can you look for translocation on a previous video? A few people are asking for it, but some please try and look for it so I can. If I have more time at the end, but I haven't. Well, I've got nothing else to answer. I will. Right. Transpiration. Right. Go through all the transpiration. It's a very long process. Right. So. Okay, I'm gonna do it backwards to the way the book teaches it. Because the book starts at the roots and then goes to the leaf. I think that's a bad idea. I think start with the leaf. So I'm going to say leaf here. Right. So we're going to start here. So we have our that's a root. So we're going to start here. Water enters the leaf, and osmosis moves water along the cells of the leaf. So here are some cells. So osmosis moves water across the leaf. Then what it has, here's a stomata there. Water will then evaporate into water vapor. Now by doing that, when it evaporates, it will go into water vapor and then diffuse out of the leaf down its water vapor potential gradient. So basically water evaporates and diffuses out of the leaf through the stomata, which reduces the water potential here. So water moves in there, which reduces the water potential here, which moves water in there, which reduces the water potential in here, moving water into the stem, reduce water potential. As you can get making water move up. So evaporation diffuses out of leaf, lowers water potential, water moves in, lowers water potential, water moves in, etc. Then, so we're now there. Now we get the transpiration pull. So we have two main forces. We have cohesion, which is water molecules sticking together. So that's a water molecule. They stick together. You then have adhesion, which is if this is the cellulose cell wall, it sticks. So we have them all sticking together. But you also get them sticking to the cell wall. That's adhesion. And this creates tension in the water. So you can get a column of water, it all just joins together like that. So you get water sticking to the cellulose cell wall and sticking to each other, creating tension, which will create a column of water. But then the water potential that up here is the lowest, so it moves up the stem. So that's how we get it moving up the stem. Cohesion, adhesion, tension, sticks together and moves up the stem in a column of water. Do you understand that so far? So we've done all that, we're now at the roots. Do you understand the leaf bit and the pull of the water up the stem? Were the two people who asked that. Right, great. So we're now in the roots. Again, we start off, water's moving up, so we're losing water here. So we're now going to have water move into the roots. Now, how does that happen? I'm just going to leave that there. Well, we have a collection of cells. We have the root cell, that's a bit of a big cell, nucleus, 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 and then we have the Casparian strip. I'm going to leave that there. Another layer of cells, stem. All right, so this is the stem, this is the root. Now, we know that water will enter the root. Oh, one second. Let me just draw another important thing. Vacuole, 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 vacuole. Now, water enters the root by osmosis, as we know, because we've got water leaving here and moves up here. Water potential is lowered here. Water enters, you know. Now, there are three paths that water can take. We've got the the apoplast, the symplast, and the vacuola. Now, the apoplast pathway is when water goes through the cell membrane, or the cell wall, sorry, not the cell membrane, the cell wall. Because there's many water-filled spaces between the cellulose molecules and water can move between them. We then have the symplast pathway, which when it goes through the cytoplasm, between gaps in the cell is called plasmodesmata. So it moves, there's another one there. And we find out the vacuolar pathway, which is exactly the same as the symplast pathway, but it just goes through vacuoles. So the three: apoplast through a cell wall, symplast through cytoplasm, and vacuola through the vacuoles. But then we have something called the Casparian strip, which is that. Now, the Casparian strip is waterproof and prevents water going through it. And what it does, it's the cell wall which is waterproof. So the apoplast pathway stops. It cannot go through the cell wall, but it can still go through plasmodesmata. So basically, it must go through the symplast or the vacuola. So what that means is you've got water leaving here, so water potential reduces, goes out that way, causes water to move throughout the root. Water goes into the root, travels three ways: apoplast through the cell wall, symplast through the cytoplasm, vacuola through the vacuoles till it reaches the Casparian strip, which has waterproof walls, meaning that the apoplast pathway must stop and it must go into the symplast or vacuola and then go all the way to the stem. So that's exactly what happens in transpiration. Does that answer your question fully, Aubrey? I hope so. Right, Jessica. Now, this is a confusing thing. I do understand, and it's got me a while. So, what you're basically saying is that, right, the inspiratory reserve volume below and the expiratory volume above should be the other way around. Now, at first glance, yes, you would totally think that, but it's what is the way the drum works. Oh, well, I'm sorry. One sec. What is the strip thing? It's annoying thing about having a lag. Oh dear, what have I done? Sorry, I've knocked my camera. Not my camera. This is awful. Sorry. What is the Casparian strip? Right. The Casparian strip is just the kind of it's waterproof. It's basically got waterproof cell walls, so it makes sure that all the water must go through the cell cytoplasm. Yeah. That's all it is. It's just waterproof walls making sure the water goes through the cytoplasm or vacuole. That's all it is. Anyway, right. What you think of it is, though, is that the way we have it, we have this drum. I'm actually gonna kind of show it weirdly. Right. Let's say this is the pen. We know that when you breathe out, air enters the tank and it rises. So here, you breathe out, and it'll go. Oh, sorry, I've done. Wait, no, ignore that. That's awful. No, this is quite hard to do. Right, I'm gonna draw it like this. Right. So you breathe out, and it goes up. Oh, like that. You then breathe in, and it goes down because it's reducing in height. You breathe out, in, out, in. So basically what it means is this is breathing, right? Because when you breathe out, it goes up, and the pen stays stationary. So that's breathing out. That's breathing in, I believe. Let me just check. No, no. No, that's wrong. Is it? Oh, I actually hate this. Hate this thing. It is so counterintuitive. Sorry. Wait. No, it isn't. That's wrong. Sorry. Again, it really makes me think this. Sorry. It's attached to the lid. So when you breathe in, it causes the lid to go down, so the pen goes down. That's you breathing in. Then when you breathe out, the tank increases, the amount of gas in the tank increases, so it gets pushed up with the pen. Breathe in, breathe out, breathe in, breathe out, breathe in, breathe out, etc. So I'm dreadfully sorry for that. Basically means breathing in is that way, breathing out is that way. So the expiratory reserve volume is the extra amount of air you can push out. So breathe more out. The inspiratory reserve volume is the amount you can breathe in. So down. So it's basically because if you look at the tank, if you breathe out, more air fills the tank, so the tank rises, so the pen goes up. If you breathe in, less air is in the tank, so the tank decreases and falls, so the pen drops as well, so it goes down. So breathing in is going down, breathing out is going up. That's why it basically is. If you look at the tank, as it goes up, pen goes up, that's when breathing out. So it's kind of just the opposite of what you think. I hope that answers your question. Was it? Yeah, I think Jessica, you answered that, didn't ask that, sorry. So can you just say if that did kind of help? If not, I'll try explain it another way. It is quite confusing. Yeah, sorry about that. It is counterintuitive. It's basically because as you breathe out, the tank rises. Okay, great. It is a confusing topic. And there you go. That's all my questions done now. Right, come on, ask some more if you want about anything. Um, ooh. Call me Jay. Yes, if we breathe in, yeah, we reduce the volume of gas in the chamber. Yeah. And would you be able to go over fetal hemoglobin? What do you mean by go over fetal? Do you mean just the difference between it? Because I'll go over. Yeah. Can you just clarify what you mean by fetal? Do you mean just the difference between fetal hemoglobin and normal hemoglobin? What do we need to know about stem cells? Could you go over the Bohr shift, which is fetal hemoglobin? Is that what? Yeah. Okay. I'll go over that then. What do you need to know about stem cells? Is that they can differentiate to form any other type of cell. They are undifferentiated, unspecialized cells. I don't know how much you do need to know actually. I don't think you need to know. You need to know what a stem cell is. I don't know. Let me have a look. Right. Right, you need to know stem cells have the ability to divide and develop into any one of several different types of cells. Young embryo embryonic cells can turn into any type of cell. They are described as totipotent. Other stem cells found in small numbers in adult tissues, like in bone marrow, are pluripotent, meaning they develop into several types of stem cells. So that's what. So embryonic ones are totipotent, they turn into anything. Adult ones found in bone marrow are pluripotent, which means they can turn into most or a number of different types. Blue flower, there is essentially not much. You do not need to know that much. So totipotent, pluripotent, embryonic, adult, that's about it. And that they can differentiate into any other type of cell. Right, Bohr shift. Right. S-shaped curve. We know this, so this is pp of O2, percentage saturation. That's a terrible T. Right. Basically, firstly, the point, fetal hemoglobin has a higher affinity for oxygen. That means, I'm going to draw down a red pen actually. Right. It basically means at a lower partial pressure of oxygen, it will be able to pick up the same amount as adult hemoglobin. So, yeah, that's basically the Bohr shift, is that little shift. There. Now, the reason why is more, if you just know the reason why, it's kind of obvious. It's because, right, this is going to be a really bad, this is the body of a mother. That is the womb with a baby in it. Little baby. There. That is your mother. It's the worst drawing of a human ever, I know. Mother, womb. Now, you have the lungs, and you have the heart. This is so bad. So, the heart picks up oxygen from the lungs and then pumps it around the body. Now, the important thing is, now it can be pumped, I don't know where exactly it gets pumped in the order, but it will, by the time it's reached the womb, it will have used some of the oxygen. Some of the oxygen will have been taken out, which means let's say, I'm making this figure up completely, but let's say you can pick up 4 oxygen molecules in hemoglobin, and by the time it gets to the womb, all hemoglobin molecules only have 3. So you basically got less. You've got 75% of the original amount. Now that means the baby needs just as much oxygen as the mother does. Each cell requires the same amount of oxygen or the baby will die. So it has to get the same oxygen requirements as the mother does for each cell, but it's got less amount of oxygen to do it in. So what it needs to do, that means it's got a lower partial pressure of oxygen, but needs to pick up the same amount. So its hemoglobin is able to pick up the same amount of oxygen in a lower partial pressure. So that is why, it's because it's got less oxygen to deal with. And that's the Bohr shift. It means it will shift to the left. Oh no, that's not the Bohr shift. No, that's something else. Well, it's part of the Bohr shift. It's it is movement. That's just fetal hemoglobin. Learn something new every day. The Bohr shift. Right, the Bohr shift is this. Right, sorry about confusion there, but I hope that answers the one about fetal hemoglobin. The Bohr shift is, you have that. Is that hemoglobin has a higher affinity for carbon dioxide than oxygen, which means as the concentration of CO2 increases, it means a higher partial pressure of oxygen is needed to be able to pick up the same amount of oxygen. So yeah, that's simply it. So as concentration of CO2 increases, it means the partial pressure of oxygen is needed to increase to be able to pick up the same amount of hemoglobin. That's all it is. So basically, as carbon dioxide concentration increases, it becomes harder to attract oxygen. Right. If in facilitated diffusion, how do carrier proteins work if ATP isn't used? Right. Okay, I'm going to be completely honest. I actually don't know why they don't need ATP. I mean, logically they would, but I think it's just that's how these proteins work. They can be, from what I can see, it's to do with opening and closing. Yeah, actually, I honestly do not know. It's something to look up online. I'm sorry about that, Jessica. I just don't know. Oh, so sorry about that. It just shows I do not know everything, which is not good. Right. Lymphatic system and lymph. Basically, you just need to know lymph is what happens when the tissue fluid is drained into the lymphatic system. Um, the lymphatic system contains a number of vessels that are similar to capillaries. They start in the tissues and drain the excess fluid into larger vessels and eventually meet in the chest cavity. And you have lymph nodes, which are kind of like glands all throughout your body, which can help produce lymph. And lymph basically contains lymphocytes. It's part of the which are produced in the lymph nodes, and these are swellings that are found at intervals along the lymphatic system. They filter any bacteria and foreign material from the lymph fluid. So it's basically kind of part of the immune system. That's what you need to know. The tissue fluid drains into it drains into the lymphatic system, which will go around the body and basically contains lymphocytes and is part of the immune system. An interesting fact about the lymphatic system and lymph nodes. I know the lymph nodes are swellings because I've currently got swollen lymph nodes. I've got something wrong with me, which means I've got swollen lymph nodes, which are, if you've ever felt, you can have them just above your kind of, yeah, there's mine. Mine are a bit below my ear, near my jawline. So that's where your lymph nodes can be. They can be all down your kind of jawline. Right. What are the limitations of photometer experiment and ways of improving the experiment? I hope that answers your question anyway. Oh, photometer. Oh, God, photometer. Limitation of photometer experiments, simply they don't actually ever work. Photometer experiments are the worst. Now, basically, limitation of photometer is stuff that it can like generalize the answer. I will get you a better answer for that. One sec. Right. Well, in the surface area of the leaves are different. You can have different numbers of stomata. So what you want, so different number of stomata mean that transpiration will increase or decrease for different leaves. That's not good. So you want to have similar sized leaves. One second. you have to assemble the pedometer underwater. So if the leaves get wet, that can reduce the water vapor potential gradient. So that means you need to assemble. You need to assemble it underwater, but not get the leaves wet. That's a way you can improve it. And also, it's quite hard to keep conditions constant. Let's say the sun came out, or temperature increased, light increased, which increases rate of transpiration. So it's quite hard to get exact conditions and everything standard, like leaf size and number of leaves in each time you do the experiment. So basically, you just need to try and control it, get it in perhaps, yeah, you need to cover, perhaps in like an oven, not an actual oven, but something that can control temperature, a walk-in oven kind of, control temperature, make sure all the leaves are the same size if you can, make sure the leaves aren't wet, things like that. What do you mean the different units and its conversion for studying cells? Can you be a little bit more specific on that? Can you explain an ECG trace, page 57? Right. You have a PQRST wave, and so, no, it doesn't. So what you mean? Doesn't explain very well. Right. Well, basically, you have P, the QRS complex, and T. And if you look at it, I explained ventricle, atrial systole, ventricular systole, and diastole. The P part of the wave is atrial excitation of the atrial systole. QRS equals excitation of the ventricles or ventricle systole, and T is diastole. So if you just know that P is atrial systole, QRS is the ventricular systole, and T is diastole, and look at what they are, then you will understand it. So I hope that makes sense. What is PR interval and ST segment? Um, P2R interval, P2R, you get the relaxation of the atria and the contraction of the ventricles. And between S and T, you get the relaxation of the ventricles. How we convert meters into right. Right. So a meter is 1 times 10. A meter is a meter. A centimeter is 1 times 10 to the minus 2. Let me just put meters. Meters. Millimeters is 1 times 10 to the minus 3 meters. Micrometers is 1 times 10 to the minus 6 meters. And nanometers is 1 times 10 to the minus 9 meters. That means a millimeter is 1,000, because 1 times 10, because if you times a millimeter by 1,000, you get a meter. A micrometer is 1,000th of a millimeter. So if you times that by 1,000, you get a millimeter. If you times it by a million, you get a meter. A nanometer is 1,000th of a micrometer. So if you times it by a million, you get a millimeter. And if you times it by a billion, I think you get a meter. One sec. Daniela. I'll talk about the different lines in a minute. But yeah, so it basically means if I said right, basically all you need to do is you need to get conformity. So if we have a cell which do I have a I don't have my ruler with me. I'm going to make up and say that is. I literally have no idea. It's going to be 25 centimeters. That's not 25, 15. Let's say 15 centimeters. I really don't know. The actual width of it is 40 micrometers. Now, what you need to do is to work out the magnification. You know that AIM. So you want to do image size over actual size. So in this case, 50. This size over that size. Sorry, you can't see it. This size over that size. You need to convert them into the same units. And basically what you do is you have 15 times 10 to the minus 2, because it's centimeters, divided by 40 times 10 to the minus micrometers, so 6, which if we get my cameraing is really bad. So 15 times 10 to the minus 2 over 40 times 10. Oh god, I can't type. 40 times 10 to the minus 6. You have a magnification of times 3750. So just basically remember that centimeters minus 2, minus 3, minus 6, minus 9, that. And then whatever value you get, if it's in centimeters, you put times 10 to the minus 2. If it's millimeters, times 10 to the minus 3, etc. So I hope that makes sense. How come, right, different images on, where is it? Right. Well, basically they're showing different things. I know, this is kind of right. So the bottom line is the electrical activity. That's the ECG. The next line is volume. And it's the ventricular volume. So it basically shows the volume of blood in the ventricles. So it starts off high, it's pushed out, so the volume decreases. So as you can see, at the QRS is when the ventricles contract, so you get blood in them, so it pushes blood out. And at diastole, it relaxes and you start getting blood flowing back in again. That's that one. The next one above it is ventricular pressure. So it's just basically showing the pressure at the different sections. So we have atrial pressure, aortic pressure. Yeah, it's showing at certain point on the PQRS. So let's say at RS, the pressure in the ventricles increases because it's contracting. But the pressure in the atrial decreases because they're not contracting anymore, they're relaxing. So yeah, that's kind of what it means. It's basically showing the ventricle volume, the atrial pressure, the ventricle pressure, and the aortic pressure at different points during the PQRS wave. So it shows you what's happening at each stage at the PQRS wave, or PQRST. I hope that makes sense. Hi, Brian. Shame not getting as many viewers as I have before. I'm averaging between 5 and 10 today. I'm assuming that's because I'm only doing AS Unit 1, perhaps. This is an AS Unit 1 biology live stream. Brian, sorry. Oh, yeah, I haven't got the book with me. Yeah, I just suggest watch my green chemistry video for that. Have you watched my green chemistry video? Because it should be on there. I'm not doing a chemistry one because I really don't have time. I've got my own A-level exams as well. Yeah, I guess I'll go over translocation then, since not got any other questions. Might as well. Right. Right. Translocation. So, phloem sieve tube, companion cell, really badly drawn. Right. Now, H plus ions are pumped out of the cell, or the companion cell, by active transport against their concentration gradient. There's more H plus outside the cell than inside the cell, so they're pumped outside the cell through active transport, so using ATP. Then they diffuse back into the cell through a co-transporter protein, which brings sucrose with it. And then sucrose will diffuse into the phloem sieve tube. So that's the first step. So hydrogen ions are actively transported out, diffuse back in through co-transporter proteins, bringing sucrose with them. It's a co-transporter protein. Sucrose then diffuses into the phloem sieve tube, and this is a source. Now, as sucrose diffuses into the phloem sieve tube, it lowers the water potential. So water also diffuses into the phloem sieve tube. I'm doing F212 at 8 o'clock till 9:30 today. Then what this causes, high pressure and there's low hydrostatic pressure down here, which causes water to move down the pressure gradient, bringing the sucrose with it till it reaches another cell. And then this is now at a sink. Sucrose diffuses out, lowers the water potential, water diffuses out, again lowering the pressure, keeping that pressure gradient. So here, sucrose diffuses in, lowers water potential, water diffuses in, high hydrostatic pressure, low hydrostatic pressure, water diffuses down its pressure gradient, bringing sucrose with it. Then here, sucrose diffuses down its concentration gradient into the surrounding cells, lowering the water potential, water follows. That is translocation. And because water diffuses out, it reduces the hydrostatic pressure, allowing that pressure gradient to continue. That's all it is. Does that make sense? Call me Jay. I hope that does. Oh yeah, a lot of these things are actually a lot more simple than they sound. The book likes to overcomplicate things. It wants to make it look a lot harder than it is, and it isn't that hard. But then again, remember, I have done this a long time ago. I'm now doing A2 biology, which is a lot harder. So this stuff is a lot easier to explain for me now, especially because I understand it. And yeah. Also, one thing, guys, if you can kind of, advertise the 8 o'clock to 9:30 F212 live stream, see if we can get a few more people. Because it's been not a hugely populated live stream tonight. So you can tell anyone who normally watches it, advertise it on if you have Twitter or the student room or Facebook or whatever, tell your friends. See if we can get a few more people. Oh yeah, sorry. Aubrey, yes, you did say, can we go over? Yes. Brian, I will do the lungs in a minute. The next question. So xerophytes. Right, xerophytes are plants that are adapted for against water loss, and they're basically just a certain number of things you need to know that you can do. Right. Now, a few things they have is a thick, waxy cuticle layer, which reduces evaporation because it's a kind of waterproof waxy layer. You can have small leaves, like needle-shaped. This reduces the surface area of the leaves. There are less stomata, so less water can be lost. If you close this, when water availability is low, the stomata will close, which means water loss is reduced. If you have hair on the surface of a leaf, it creates a microclimate. So it means that moisture gets trapped in there, which reduces the water vapor potential gradient because you have more water vapor outside. Then generally, actually, the stuff you do need to know is just there's so much to ask, they can't ask it all. So they could ask anything in the textbook, but there's just so much they can't. Anyway, so yeah, the hairs trap water vapor, reducing the water vapor potential gradient, so there's less diffusion of water out of the plant. Pits containing stomata, so you kind of have on a leaf. You might have the leaf looking a bit like this, and the stomata will be in there. Again, this traps water vapor here, reducing the water vapor potential gradient, and so we have like, I don't know what, that's the leaf. That's the leaf there, sorry. So this is the leaf. Traps, so there's the pit, traps water vapor, so reduces the gradient. You roll the leaves again, traps air, so water vapor potential gradient released. And densely packed spongy mesophyll reduces the cell surface area. Now, it's exposed to the air inside the leaf, so less water can evaporate. So if you have, these are the cells, lots of space for water to evaporate. If you have this, a lot less space for water to evaporate. Brian, yes, you can look at this live stream again. It will be posted. I don't know if it will be posted today. It's whenever YouTube decides, but it will be posted by tomorrow. So yes, you can watch it again. So yeah, those are all the different things. You have pits, hairs, and rolling the leaf to create a microclimate, trap air. You have closed stomata when water availability is low, thick, waxy cuticle, and densely packed spongy mesophyll, and reduced surface area. So yeah, I hope that makes sense. Right, process in the lungs. See, this is another thing where inspiration and expiration aren't really explained in the book very well. I believe they're very well explained in the CGP revision guide. I think is where it's done very well, but I don't have that with me, which is a bit annoying. Yeah, it's not actually explained in the book at all. Not very. Oh no, it is. But oh yeah, right. Inspiration. Your diaphragm contracts. So basically, one sec, let me get rid of that. Right. So diaphragm contracts and becomes flatter. And what this does, diaphragm flattens. The external intercostal muscles contract to raise the ribs, so kind of they go out that way, which basically means the volume is increased. If the volume is increased, the pressure will decrease, which causes air to flow in. So diaphragm contracts to go flatter. External intercostal muscles will contract to raise the ribs, increasing the volume of the chest, causing pressure to decrease, air to flow in. That's inspiration. Expiration, the opposite happens. Diaphragm raises. The external intercostal muscles relax and the ribs fall, which makes it look like a sad face. The volume of the chest decreases, which raises the pressure of the lungs, which causes air to be pushed out. And also at the same time, you have the elastic fibers in the alveoli. They suddenly contract as well. Not contract, they recoil, which helps push air out as well. So yeah, I hope that makes sense. So inspiration, everything contracts, increases volume, decreases pressure, air flows in. In expiration, everything relaxes, so volume decreases, pressure increases, air pushed out, and there's also recoil of elastic fibers to help. So I hope that answers your question, Brian. Call me Jay. It's fine. To be honest, I don't do much in the evenings. The only thing I do in the evenings is tend to sleep and eat. So it's not really taking up that much time for me. But yeah, there we go. Since that is all the questions done, I think we are going to stop there. I know it's 7 minutes early, but I think that is all the unless anyone has another incredibly burning question they want to ask. What happens to the organs, though? Well, what do you mean? The digestive organs are just pushed down. Everything's flattened, so it's kind of pushed down further into your stomach, just increasing the volume of the chest. I was wondering where you were, James. Well, I did post a video yesterday saying there was one on, and I did create live streams a few days ago and did a video about it. But don't worry, you can stay for the. Could you email me in the future? Um, I got. Well, there won't be any more AS ones after today, so I won't be able to email. These are the last ones today. What do you want help with, James? Quickly. Yeah. Well, so they kind of get pushed down the stomach, get flattened by the diaphragm. Ways to revise. I can't have. Could you have? I can't have a whole stream on ways to revise. But if you really need to, email me munrot07@yahoo.co.uk, and I will happily go through it. I've done I've done like how to answer questions and stuff on previous live streams. If you want any help, email me with a specific question, munrot07@yahoo.co.uk, and I will answer it as soon as I can. But yeah. Microtubules, they kind of rub together, they just basically cause movement. Microtubules, if they rub together, they can push things around. So organelles can have microtubules, which when they rub together will cause movement, and then organelles can be pushed along fibers, because the whole cells contain all these like highways of fibers, which if you cause movement, they'll move along these highways. That's basically what it is. How do I revise? Right. Firstly, Aubrey, there's a live stream at 8 till 9:30 tonight for AS. There'll be an A2 live stream. I don't know if you're in A2 retaking AS or you're doing AS for the first time. That applies to anyone. But there'll be the final AS one, which is on Unit 2, will be tonight, 8 till 9:30. A2 ones will be at on Monday for Unit 4, and then half term for Unit 5, and a further one for just general unit for general A2. How do I revise? I revise very boringly. I just create pages upon pages of notes. I haven't got my notes with me, but I make pages of black and white, boring notes, occasional diagrams. Then I use this book to answer all the questions in it. So here's all the pages I've done for answering various questions. So I answer all the questions from in here, like there. Once I've answered all these questions, I've made all the notes and all the topics, answered all the questions from that book. I then have a pack of exam questions, past papers, and all things like that. It's in the descriptions of any one of my videos, actual content videos. It's in there. So yeah, I will. If you can find my email in there. Anyway, I don't actually have a Facebook or Twitter page. No, I don't use it. No. I'm not that famous enough to need one. Anyway, so I make these black and white notes, I answer the questions, and I answer all the exam questions using the mark scheme carefully to look at how I've done, and anything I've got wrong, I re-revise, look at why I got it wrong, and then just kind of repeat. That's how I revise. But it doesn't work for everyone. Microtubules do a lot of things. Yes, they can also provide an anchor for proteins, but yes, they do a lot of things. How do I find studying biodiversity? Certain things are okay, certain things are sampling. No, I can't do sampling. It's the most boring thing I've ever done. It comes up again in A2. It's so annoying. Reasons maintain, like looking at actual biodiversity and reasons maintaining it, it was all right, but it was kind of obvious, and then the questions on it are awful. Then other things in the biodiversity module, so the classification, evolution, I really enjoy. I'm so mad about evolution. I love evolution a lot. So that bit was actually all right to study. Well, there's my ruler. So the biodiversity the actual biodiversity, looking at sampling, measuring biodiversity, yes, that is awful and dreadful. But evolution and classification in that module, I enjoy them. I enjoy doing them. Okay, see you, Daniela. Do I use my notes to make videos? No, I use a text. I use all my various books to make the videos. But I use all the various books to make my notes as well. But it's just slightly better to use a textbook so I get the exact wording and make sure I haven't missed anything out. I don't want to, if I transcribe from the book to the notes, then, right, books to notes and then use notes to the video, there could be a mistake or two cropping in. If I use the books, it's a lot less likely for mistakes. So I use the books. Brian, when you said, can you write down your email? It is in my videos. Okay. If you look at any one of my content videos, go to the description. It is in there. What books do I use? I'll show you. Right. Official textbook. Ta-da. I know it's an A2 one, same I've got here. CGP textbook, use that. CGP revision guide, the OCR revision guide, and there's a Cambridge textbook as well, which I also sometimes use. Yeah, the biodiversity question, it's the rule of biology. If you have easy content, biodiversity is easy to study, there's nothing difficult about it, then you have horrendously hard questions. If you have hard content, like some of the A2 questions, you get ridiculously easy questions on that. It's unfair. The videos are incredibly similar to well, the notes I make on the videos are a lot less detailed than the notes I make actually written down, but the amount I speak is exactly the same as the amount I would have written down. I don't speak to myself to make my notes, I write them down. But on videos, I do a lot of audio stuff, so I speak rather than write it down. So my notes that I write down on the videos are a lot less detailed, a lot less in content, but the content I speak and say is exactly the same level of detail I use for my own notes. Right. Which one from the load is best? Use the textbook. If you ever in doubt, use the textbooks. They cannot fault you if you use a textbook, but use textbook and a CGP textbook. Those two together are amazing. Those two are the best. Right, resolution of microscope. Again, James, resolution of microscope. Just look at what it says here. What do you want? Electron microscope. Right, electron microscope resolution is 0.2 nanometers. That's what you say. It's in textbook 0.2 nanometers, so you say that. And yeah, I'm going to have to say now, no more questions, because I would generally be okay to stay a little bit longer, but textbook, use textbook. Which one should I trust? Always trust the textbook. When in doubt, go to the textbook, because if you copy an answer from the textbook, obviously not literally copy it in an exam, if you learn an answer from a textbook, they cannot fault you on it as long as you answer it in the right place. Anyway, I do need to say I need to go now because I'm doing another live stream in an hour and I need dinner. Because otherwise I'll be, not getting any dinner till 9:30. Actually, that's an idea. I might do a live stream after the exam, or I might do an actual video. Probably best to do a video. Thing is, with the AS ones, would you like an AS video on the exams? Because I will not be doing these exams, and I will not know what was on the. Well, actually, I will. I've got a friend who can do it, so I'll do a video. I'll do a video on the exam, but I will not do a live stream on them because that's a bit pointless. Anyway, yes. I need to go now because I do need to eat. I will see any of you who are staying for the F212 live stream at 8 o'clock till 9:30. I hope you will come. Try and get as many other people as you can to come. So tell all your friends who want to revise biology, or post it online, or whatever you want to do. And I will see you in an hour. So thank you very much, and goodbye.